Morning students, today we are starting a new chapter, that is enzymes. Okay, right? So the term enzyme is coined by Kuhni. Term enzyme. The term enzyme is coined by Kuhni. Okay. Sir, the first enzyme that was isolated and discovered is zymase. What is the first enzyme that was discovered, sir? Zymase. Therefore, first discovered enzyme. Which enzyme was first discovered, sir? Zymase. Okay, right? Sir, this zymase enzyme was discovered from where, sir? Yeast. Discovered from yeast. Sir, yeast means unicellular fungi. Yes or no? Unicellular fungi. Okay. Sir, the first crystallized and first purified enzyme is urease. What is the first crystallized and purified enzyme, sir? Urease. First crystallized and purified. First crystallized and purified enzyme is what it is? Urease. I think this I told in mineral nutrition also. What is the enzyme name, sir? Urease. From which plant, sir? From jock bean plant. It is not jackfruit, it is a jock bean plant. This jock bean plant is also called as a lobia plant. It is also called as what, sir? Lobia plant. Jock bean plant, lobia plant. Okay. Therefore, first discovered enzyme zymase. First crystallized, first purified enzyme urease. This urease was crystallized and purified from a plant by name jock bean, that is lobia plant. Okay. Sir, enzymes, they act as what, sir? Catalyst. Enzymes, they act as a catalyst. Enzymes, they act as what, sir? Catalyst. Sir, actually these catalysts are of two types. Catalyst. Okay, right? Sir, these catalysts are of how many types, sir? Two types. Catalysts are of two types. Okay, what are they? Number one, inorganic catalyst. What is the first one, sir? Inorganic catalyst. Inorganic catalyst. Second one is organic catalyst. What is the second one, sir? Organic catalyst. Okay. Sir, this inorganic catalyst works efficiently at high temperatures and high pressures. Which catalyst, sir? Inorganic catalyst works. Inorganic catalyst works efficiently. Works efficiently at high temperatures and high pressures. Sir, who is working efficiently at high temperatures and high pressures, sir? Inorganic catalyst working efficiently at high temperatures and high pressures. What is the second one, sir? Organic catalyst. What they are, sir? Organic catalyst. Sir, this organic catalyst, they are called as enzymes. Organic catalyst, they are called as what, sir? Enzymes. Sir, these enzymes are denatured at high temperatures. Denatured at high temperatures, that is above 40 degrees Celsius temperature. It means temperature is more than 40 degrees. These enzymes are denatured. They won't work. Whereas inorganic catalysts, they work efficiently at high temperatures and high pressures. Sir, organic catalysts, what they are called as enzymes, they are denatured. They are denatured. They are denatured, right, at high temperatures. They are denatured at high temperatures. Sir, here high temperature means how much, sir? Above 40 degrees Celsius temperature. 40 degrees Celsius temperature. Okay. Sir, this organic catalyst, we are calling them as what, sir? Enzymes. Sir, these enzymes are of two types. These enzymes are of how many types? Two types. First one is protein enzymes. Second one is RNA enzymes. What is the first one, sir? First one is protein enzymes. First one is what, sir? Protein enzymes. Protein enzymes. Second one is what, sir? RNA enzymes. What is the second one, sir? RNA enzymes. RNA enzymes. Okay, right. Go for the first one. Which enzymes are protein enzymes? Go for the first. What it is? Protein enzymes, right? Go for the first one. What's that? Protein enzymes. Protein, what's that? Enzymes. Protein enzymes. Okay. Now, see here. Sir, these enzymes are made up of what, sir? Proteins. Made up of what? Proteins. They are made up of proteins. Sir, proteins means what, sir? Proteins means they are polymers of amino acids. Polymers of what, sir? Amino acids. They are polymers of amino acids. It means they are polypeptides or polynucleotides. They are polypeptides. They are polypeptides. They are polypeptides. Okay, now listen to the statement. Sir, almost all enzymes are chemically made up of proteins. I am not saying all enzymes are chemically proteins. I am saying almost all enzymes are chemically made up of proteins. What is the exceptional case, sir? RNA enzymes. See here, these enzymes are chemically made up of RNA nucleic acids. Therefore, except RNA enzymes, almost all enzymes are chemically made up of what, sir? Proteins. Therefore, almost all enzymes are proteins. Almost all enzymes are what, sir? Proteins. Okay, right, sir? Except right, except RNA enzymes. Except what, sir? RNA enzymes. Remaining enzymes are chemically. Remaining enzymes are chemically what, sir? Proteins. Remaining enzymes are chemically proteins except RNA enzymes. So, almost all enzymes are what, sir? Proteins. Okay. Now, listen this statement. Sir, we are discussing about protein enzymes. Okay, right? Everywhere they won't give protein enzymes. Enzymes, we are speaking about enzymes means, right, they are what, sir? Protein enzymes. Okay, because almost all enzymes are chemically what, sir? Proteins. Therefore, right, enzymes are chemically what, sir? Proteins. They are colloidal in nature with tertiary structure. With which structure, sir? Tertiary structure. Therefore, enzymes. What? Enzymes. Right. These enzymes are almost all, I told already, what they are? Proteins. Yes or no? Right? What in nature? Colloidal in nature. Okay, having which structure? Tertiary structure. Having which structure, sir? Tertiary structure. But take isoenzymes. Which enzymes, sir? Isoenzymes. Sir, isoenzymes means structurally different, functionally similar. For example, take lactate dehydrogenase. Lactate dehydrogenase, it's a isoenzyme. This isoenzyme is having quaternary structure. Having which structure, sir? Quaternary structure. In first year, right, we read structure of proteins, primary structure, secondary structure, tertiary structure, quaternary structure. Primary, secondary, tertiary, quaternary. Sir, those are structures of protein, no sir. Here we are discussing enzyme, no. Just now I told almost all enzymes are chemically proteins. Okay, therefore enzymes, they shows which structures are tertiary structure, except which enzymes? Isoenzymes. Sir, isoenzymes, right, they shows quaternary structure. Isoenzymes means structurally different but functionally similar. Structurally different, functionally similar. Okay, right? Which enzymes are isoenzymes? Isoenzymes. Sir, isoenzymes means what, sir? Structurally different and functionally similar. Structurally different, functionally similar, they are isoenzymes. Sir, these isoenzymes, they have which structure, sir? Quaternary structure. They exhibit which structure, sir? Quaternary structure. Sir, these isoenzymes, best example is right, lactate dehydrogenase. What is the enzyme name, sir? Lactate dehydrogenase is an example for isoenzyme, right? Now listen here. Just now I told these proteins, they shows a tertiary structure. Yes or no? Right? Now, sir, a protein in tertiary structure bends along its own backbone. What is bending along its own backbone? The protein is bending along its own backbone. In which structure? Tertiary structure, and forms many crevices or pockets. See here, sir, this is one crevice or this is one pocket. Like this, many pockets are formed. When, sir, when this protein, what, sir? When protein, right, when it bends along its own backbone, when protein bends along its own backbone, when protein bends along its own backbone, in which structure? Tertiary structure. Okay, what is bending? Protein is bending along its own backbone. In which structure? Tertiary structure. What are formed? Crevices or pockets. How many? Many pockets are formed, or many crevices are formed. One of such pocket is named as, active site. One of this pocket is named as what, sir? Active site. Sir, when substrate comes and binds in this active site, what, sir? Active site, then enzyme carries out the reaction. Therefore, right, when an enzyme is doing a reaction, when it is going to carry out a reaction, when substrate comes and binds in the active site of an enzyme. Okay, when substrate comes and binds in the active site of an enzyme, then this enzyme will carry out the reaction. When the enzyme, okay, carries out the reaction, when, sir? When substrate comes and binds in the active site of an enzyme. It means to carry out a reaction, an enzyme requires what? Active site. Enzyme is getting that active site when it attains which structure? Tertiary structure. Enzyme gets this active site when it attains which structure, sir? Tertiary structure. When it attains which structure? Tertiary structure. Right? Right? One such pocket. Many pockets are formed, no? One such pocket. One such pocket is called what, sir? Active site. Which site, sir? Active site. Okay, right? Enzyme. What, sir? Enzyme. Enzyme carries out. Enzyme carries out a reaction when substrate comes and bind where in the active site of an enzyme. When substrate comes and binds in the active site of an enzyme, then enzyme is going to carry out a reaction. Then it is going to carry out a reaction. Yes or no? Right? Therefore, what has to come and bind? Substrate has to come and bind in the active site of an enzyme. Okay, right? Therefore, enzyme, what it requires? Enzyme requires active site. Okay, to carry out a reaction. Right? Therefore, so enzyme requires, what it requires? Enzyme requires what, sir? Active site. Sir, when an enzyme will get the active site, sir? Right? So enzyme requires what? Active site which is attained. Which is attained. It means when this active site is attained, which is attained right in which structure, sir? Tertiary structure. Which is attained in which structure, sir? Tertiary structure. Just now I told, sir, enzymes are denatured at high temperatures, that is above 40 degrees Celsius temperature. But there are some organisms which can survive at high temperatures, that is 80 to 90 degrees Celsius temperature. These organisms are thermophilic organisms. They are growing in hot vent sulfur springs. Therefore, sir, if you isolate an enzyme from these thermophilic organisms, that enzymes are tolerating high temperatures, because organism itself tolerating high temperatures. So the enzymes isolated from such organisms can tolerate high temperatures. Therefore, thermally stable enzymes can be isolated from thermophilic organisms. Okay, in biotechnology, that is in PCR, okay, we read about one enzyme, Taq polymerase, a type of DNA polymerase, isolated from thermophilic bacteria by name Thermus aquaticus. Yes or no? That Taq polymerase is a thermally stable enzyme. Therefore, thermal stability is an important quality of such enzymes isolated from thermophilic organisms. Right? Now listen this statement. Sir, we are discussing about organisms that are growing in hot vents and sulfur springs. Yes or no? Right? Sir, organisms growing right in hot vents, sulfur springs. What are these organisms called as thermophilic organisms? Thermophilic organisms. What are these organisms called as thermophilic organisms? Sir, from these thermophilic organisms, from which organisms, sir? Thermophilic organisms, right? Enzymes isolated enzymes isolated, right, can tolerate 80 to 90 degrees Celsius temperature. 80 to 90 degrees Celsius of what, sir? Temperature. Therefore, sir, enzymes isolated from these thermophilic organisms, that enzymes, they can tolerate 80 to 90 degrees Celsius of temperature. It means these enzymes are thermally stable. So, enzymes isolated. So, enzymes isolated from what? Thermophilic organisms. Enzymes isolated from which organisms, sir? Thermophilic organisms are thermally stable. They are thermally stable. Okay, right. Thermal stability. Thermal stability of such enzymes means enzymes isolated from thermophilic organisms. Thermal stability of such enzymes, okay, is an important quality. Is an important quality of enzymes. Therefore, the enzymes which are isolated from thermophilic organisms, such enzymes are thermally stable. This thermal stability is an important quality of such enzymes which are isolated from where, sir? Which are isolated from thermophilic organisms. Therefore, thermal stability of such enzymes is an important quality of enzymes, which are isolated from which organisms, sir? Thermophilic organisms. Okay, sir, thermally stable enzyme, best example, Taq polymerase. Yes or no? What, sir? Thermally stable, right, enzyme. Best example is what, sir? Taq polymerase. What it is? Taq polymerase, which is a type of DNA polymerase used in PCR. Used in what, sir? PCR. That is a polymerase chain reaction. Right? Right. Now, see here. Go for the second one. What, sir? RNA enzymes. Which enzymes, sir? RNA enzymes. Sir, RNA enzymes, also called as a ribozyme. Also called what, sir? Also called ribozyme. Therefore, ribozyme means what, sir? RNA enzymes. It means here RNA is acting as an enzyme. Yes or no? RNA acts as an enzyme. Sir, RNA is a nucleic acid. It means these are nucleic acid enzymes. Yes or no? Therefore, RNA acts as an enzyme. Acts as an what, sir? Enzyme. Sir, but this RNA is what, sir? Nucleic acid. RNA is a nucleic acid. It means a nucleic acid is acting as an enzyme. Sir, this RNA enzymes, nothing but RNA is acting as an enzyme. RNA means what, sir? It is a polymer of ribonucleotides. Yes or no? Therefore, polymer of what? Polymer of ribonucleotides. Polymer of ribonucleotides. It means it is a what, sir? Polynucleotide. Yes or no? It's a polynucleotide. Which nucleotide? Ribonucleotide. Okay, sir, we are discussing about RNA enzymes, that is ribozyme. Yes or no? Sir, the first discovered RNA enzyme is L19 RNase. What, sir? L19 RNase. L19 RNase is the first, right? It is the first discovered, it is the first discovered RNA enzyme. The first discovered RNA enzyme. Sir, this L19 RNase was first discovered RNA enzyme. Who discovered it, sir? It was discovered by a scientist by name Ketch. By name what, sir? Ketch. By name Ketch. What is the scientist's name? Ketch. Therefore, L19 RNase first discovered by Ketch. Where, sir? In a organism by name tetrahymena. Sir, this tetrahymena, it is a protozoan. Sir, protozoan means comes under kingdom Protista. Protista means it's a eukaryotic or prokaryotic kingdom. It's a eukaryotic kingdom. Therefore, first discovered RNase enzyme is what, sir? First discovered RNA enzyme is L19 RNase. It was discovered by Ketch in an organism by name tetrahymena, which is a protozoan. That is a Protista, comes under Protista. It's eukaryotic. Right? Later, a scientist by name Altman. Later, right? Altman. What's the scientist name? Altman. Right? Altman discovered another RNase enzyme. Another RNA enzyme by name RNase P. RNase P is also called as ribonuclease P. Okay, therefore later Altman discovered another RNA enzyme. What it is? RNase P. RNase P is also called as ribonuclease P. Okay, sir, this enzyme was discovered by Altman, right, from a prokaryotic organism. From a prokaryotic organism. From a prokaryotic organism. Okay, right? Sir, we are discussing about RNA enzymes. Sir, another example for ribozyme. Till now we got two examples for ribozyme. Number one, L19 RNase. Second one is RNase P. Now go for the next example. Next example for ribozyme is 23S rRNA, 23S rRNA and 28S rRNA. So 23S rRNA, right, present in larger subunit of 70S type of ribosome. Where it is present, sir? In first year we read this statement. Present in larger subunit. Present in larger subunit. Present in larger subunit of which ribosome, sir? 70S type of ribosome. In 70S type of ribosome, what is a larger subunit called as a 50S? Whereas this 28S rRNA, where this is present in larger subunit. Present in larger subunit. Present in a larger subunit of 80S type of ribosome. Present in larger subunit of 80S type of ribosome. Sir, what is a larger subunit of 80S type of ribosome, sir? 60S. Yes or no? Sir, this 23S rRNA and 28S rRNA, these two enzymes are involved in the formation of peptide bond. These two enzymes are involved in the formation of peptide bond. These are peptidyl transferase enzymes. These are which enzymes, sir? Peptidyl transferase enzymes. Therefore, who is acting as peptidyl transferase enzyme? 23S rRNA, 28S rRNA. Therefore, peptidyl transferase is an example for ribozyme. Peptidyl transferase is an example for ribozyme. It means peptidyl transferase is chemically made up of ribonucleotides, polymer of ribonucleotides. Okay, therefore, sir, this peptidyl transferase example for ribozyme. What are the function of peptidyl transferase? Involved in formation of peptide bonds between amino acids. Peptidyl transferase helps in formation of peptide bonds between amino acids. Helps in the formation of peptide bonds between amino acids. Okay, right? Thank you.